Good morning, dear students. We are going to discuss nervous system histology today. The basics of nervous tissue we have already discussed in our previous lecture. Structure of neurons, neuroglial cells, everything has been taught. I just kept the slides so that you will have review in your mind. Now today we are going to discuss in detail about three slides, that is cerebrum, cerebellum and spinal cord. These are the three slides which you will be seeing under the microscope, and you are supposed to draw a well labeled diagram and write the points regarding these three slides, that is cerebrum, cerebellum and the spinal cord. Now when you see this cut section of the brain, you will be seeing the cut section of the brain will be showing outer gray matter and inner white matter. The innermost gray matter masses are also seen, which are called as basal ganglia. The details of this you will be studying when we do neuroanatomy. Gray matter is known as the cortex, and it is 1.5 to 4.5 mm in thickness, convoluted in the sense it is highly folded in man, so that the surface area of the brain is highly increased. It is quite smooth in lower animals. Now, as it is histology, we are going to just enlighten our knowledge on what are the histological features when you just take a biopsy or autopsy specimen and prepare this slide. How does the cerebrum look? The cerebrum has 6 layers in the cortex region. The first layer is called as plexiform layer, also called as molecular layer. It has mainly dendrites and axons and horizontal cells of Kajal. Then second layer is outer granular layer, where we see small pyramidal cells and stellate cells. Stellate are star-shaped cells. Third layer is pyramidal cell layer. This is quite important layer, which has pyramidal cells of moderate size. Fourth layer is ganglionic layer, where we have large pyramidal cells, also called as Bed's cells, B-E-T-Z, bed cells, satellite cells, and cells of Martinotti. The last one is the multiform layer, where you see all type of cells and fusiform cells. Okay, so that is like these are the 6 layers. Okay, then this is a diffuse atlas picture what you are seeing, and we are showing you here the layers like the molecular layer, external granular, external pyramidal cells. You can see these are the pyramidal cells which are black in color. They are little smaller in size. This is internal granular layer, and these are large pyramidal cell layer, where you have size of pyramidal cell is quite big. Then we have multiform layer. Okay. See here now. So if like these are the 6 layers which we have to be thorough with. Okay, the molecular, external granular, pyramidal, internal granular, ganglionic cell layer, multiform layer. Third and fifth layer are characteristic and contain pyramidal cells. These are the cells which make the cerebrum very much unique, and which is the seat of intelligence, very, very important layer. Okay, so neuroglia are of course disposed in all layers. We have about 10 billion neurons in cerebral cortex, and because one neuron can establish synaptic connections with as many as 1 lakh others. White matter contains interlacing bundle of myelinated nerve fibers having no neurolemma sheath. In HND stain, cerebrum shows pale color, almost structureless appearing background, known as neuropil, and consists of framework of naked nerve fibers and processes of neuroglial cells. Dark round nuclei of oligodendrocyte lie close to the white matter. Nucleoli of astrocyte nucleus are not prominent as those of neurons. Motor and premotor areas are characterized by agranular cortex, while sensory areas are characterized by granular cortex. Okay, this is HND stain, which is not so much clearly visible. Okay, these are silver impregnation methods which gives a better clarity of these layers of cerebrum. We can see this pyramidal cell. Okay, these are interneurons. We have molecular layer. Uppermost covering is the pia mater, which is one of the meningeal layer of the brain. Okay. You can also see this is one more silver stain picture, where you can differentiate layers better when you compare with H&E slide. Okay, you can see the size of pyramidal cells here. Apical dendrites, we can see. This is neurofilm. Once you are done with cerebrum, we have to check with cerebellum slide. So cerebellum, here also if you observe, it is highly folded, forming so many like like you use the word salsagere usually in the cerebrum, and but here the folds are much denser when you compare with the cerebrum, concentrating a large number of area within a small surface area. Okay, then that's why it is called a small brain with big functions. So cerebellum is serving to coordinate voluntary movements and maintains normal posture and equilibrium. It has outer gray matter as you can see here and inner white matter. Okay, deep inside this white matter again you are finding this gray matter which are called as cerebellar nuclei. Grossly it is showing so many folds. We use the word folia for this, not salsagere. Okay. Now, histology of cerebellum. See, this outer area is cortex and inner area is medulla. Okay. Cortex, it forms deeply convoluted folds called folia. These are the folia, as you can see here, these deep folds. Okay. It has central core of white matter containing 4 pairs of nuclei. This is what I have shown you. Okay. Then so the nuclei are named as dentate nucleus, emboliform, fastigious, and globosus nucleus. Dentate, emboliform, globosus, it's labeled here, and fastigiae nucleus. These are the cerebellar nuclei. We will again elaborate these nuclei when we do gross anatomy of cerebrum. Now this is histology. We will confine our explanation only to the required context. Okay. So now when you go to the histology of cerebellum, when you look under the microscope, cerebellar cortex will show 3 layers. This outer layer which is shown with green arrow is molecular layer, which is most superficial. The one which is shown with red arrow, that is Purkinje cell layer. And the deeper one inside you have is granular layer. Okay. Then the cortex, as I said, you can actually remember like a code MPG. From outside to inside, molecular, Purkinje, and granular. So outermost is M, P, and G. Outermost is molecular. It stains light with eosin. Again, we have to go with silver impregnation methods only. H&E is very weak in staining nervous tissue, neural tissue. Middle is Purkinje cell layer. This one. This you can see the Purkinje cells. Inner layer, this granules you are finding. This is the granular layer which stains intensely with hematoxylin. Okay, this is high magnification where you can see MPG layers. This is molecular layer. Okay, interspersed with basket cells. This big cell is Purkinje cell, which is a specialized cell of cerebellum which is responsible for maximum functions of cerebellum. Like we have pyramidal cells in case of cerebrum. Similarly, the specialized cells of cerebellum are Purkinje cells. And these cells, whatever we are finding below, are granular cells. Okay. Here we have Golgi type 2 cells, granule cells, and the glomeruli. Now let us elaborate these MPG layers. First layer I said is molecular layer, which relatively contains few neurons and large number of unmyelinated fibers. Show small cellate cells and basket cells in deep stratum. Axons of basket cells form network around the cells of like something like this Purkinje cells. Red in color are Purkinje cells. Okay. Then in between I have shown you here basket cells, right? Okay, which we find in the molecular layer. Then axons, one basket cell synapses with 70 Purkinje cells. Okay. Stellate cells are scattered near the surface. You can see this is the stellate cell. There are dendrites. Dendrite synapse with parallel fibers and axons with dendrites of Purkinje fibers. Okay. You can see this is Purkinje cell. Here we have basket cells. It will be actually synapsing with around so many 70 Purkinje cells. We have stellate cells like this. So these 3 cells are the major components of molecular layer. Okay. Of course, so many white fibers also we see. The next Very important layer. Purkinje cell itself comes as a short note or very short note. So this Purkinje cell, if you just look at, it looks like flag shape. This one, Purkinje cell, this red one is Purkinje cell. Okay. So flask-shaped neurons, which are characteristic feature of cerebellum, shows one row of Purkinje cell. As you can see here, cell bodies are evenly spaced, same centimeter space you will find between the Purkinje cells, millimeter space, sorry. Dendrites show arborization. That means this branching pattern is called as arborization in molecular layer and their spines. Let me show you. This is arborization and the spines are seen in which layer? Upper one, M, molecular layer. Okay. Then the dendrites synapse with parallel fibers, axons of stellate cells, climbing olivocerebellar fibers, and collateral basket cells, as you can see here. These are axons which are going down. The axons are only efferents which pass through granular, MPG, granular layer, and end in the nuclei of cerebellum. Okay, these are the axons, and this arborization pattern we have seen with dendrites. Okay, now the axons are coming down, piercing the granular layer, okay, medulla, the white matter of the cerebellum to reach the intracerebellar nucleus. Okay, then the last layer, this gray in color, the inner granular layer. This darkly stained contains numerous round granule cells. Okay, this one, this gray in color is the granular layer. You can see it has granule cells here. Okay, spaces not occupied by these cells are called as cerebellar islands. These islands are occupied by this area. This is glomeruli. Okay, axons of granule cells branch in the form of T, so that axons make contacts with the dendrites of Purkinje and basket cells. You can see Purkinje cells and basket cells. They go like that. These are the dendrites, these granule cells. Okay. Now you can see these are the glomeruli region which are seen in this layer. It is formed by dendrites of these granule cells and axons of Golgi cells. Okay, synapsing with the terminal like terminals of mossy fibers. These are the mossy fibers. Brush cells are also seen in the granular layer, which are monodendritic. All the intrinsic neurons of cerebellar cortex are inhibitory except granule cells, which are excitatory in function. Okay, only these are excitatory. Rest all are inhibitory. Okay, then once you are done with MPG layers, molecular, Purkinje, and granular, now let's move to this white matter which contains central core of myelinated nerve fibers. Two types of afferent fibers are seen in cerebellum. Okay, we have climbing fibers which pass through granular layer and reach Purkinje cells, dendrites in molecular layer. Mossy fibers are other white fibers which synapse in granular layer here. Okay, these green in color are climbing fibers and blue in color are mossy fibers. So key points of identification, MPG layers, you try to remember. Outer molecular layer, these are Purkinje cells, and inner we have granular layer. This is gray matter. Inside we have the white matter. Okay. Then once we are done with that, now let us see the third slide, that is the spinal cord. This is ovoid in transverse section. It contains this butterfly shaped area is nothing but gray matter, and outside we have white matter. This is the central canal. Okay. Now see here, this is the anterior end and this is the posterior end. This area is anterior median fissure. This is posterior median sulcus. This is the anterior horn of gray matter. This is posterior horn of gray matter. This is lateral horn, which is clearly seen in the thoracic part of this spinal cord. This is central canal, which is lined with ependymal cells, where we have CSF moving. Rest all is the white matter outside. Okay. And of course, it has three coverings from outside to inside. If you see, outermost is dura matter, middle is the arachnoid matter, and innermost is the pia matter. Whatever round structures we are finding, these are nothing but the blood vessels. We have anterior spinal artery and vein. We have two posterior spinal arteries and veins. Okay, many set of veins we will see all around. Again, this is silver impregnation. Special stain is used. This is the central canal. Okay, this is anterior horn. You can see anterior median fissure. This is posterior horn. This whole area dark brown in color is white matter. Light yellow in color, whatever area here is the gray matter. Okay, beta. This is anterior horn. This is posterior horn of the gray matter. Okay, this is all white matter. And of course, surrounding we have the meninges. Gray matter is in the form of right and left columns, right? You can see this one. This is right column, this is left column connected by this area. In between, you will have the central canal. Connected with each other in the middle, giving H-shaped appearance in transverse section. Anterior horns are broader and shorter and do not reach the surface of spinal cord. See, these are shorter and broader and they are not reaching the surface. Whereas posterior horns are narrow, longer, they are reaching the surface of the spinal cord, right? That is. Lateral horns are like very small. These are the lateral horns, maybe or may not be present. Okay. In the thoracic and upper lumbar segments only, we find these lateral horns and their cells are connected with sympathetic fibers. Now, the gray matter contains nerve cells, nerve fibers, neuroglial cells, and blood vessels. Neuroglia are arranged in sponge-like network except at two places where they are condensed, one at the tips of the posterior horns and others surrounding the central canal. Rest all places they are actually very much sponge-like. The nerve cells, this is the high magnification. You can see this is the anterior horn. We have multipolar neurons, variable in size. We have Golgi type 1 cells. Okay. I have long axons which pass through the white matter. Golgi type 2 cells have short axons and they do not leave the gray matter. So rest all outside what you have is the white matter consists of myelinated nerve fibers. Neurolemma sheath is absent and are cut transversely. The TS spinal cord is surrounded by three meninges, dura, arachnoid and pia matters. Okay, I hope you understood. Thank you.